Hello students, welcome to EPG Pathshala. I am Dr. Mini Singh from the Department of Biotechnology at Punjabi University, Patiala, and today we will talk about Nanobiosensors for Food Safety Part 1, particular Electrochemical Biosensors under the paper of Nanobiotechnology. We have understood now that there are certain concerns posed of food safety, whether it is food infections, whether it is food contaminations, or so on and so forth. These can be classified into pathogenic detections or chemical residues or fraudulent chemical detection. The two most important kinds of biosensors which are being developed for making ultrasensitive biosensors are the nanobiosensors with electrochemical transducers and those with optical transducers. In this module, we will understand the concepts behind electrochemical transduction. The reason why electrochemical transducers are so successful in the development of biosensors is because of their specificity of response and their ultrasensitive detection probability. Based on this module has been devised with the following objectives. First, we will talk about food safety risks very briefly. Then we'll talk about food analytics, the need for ultrasensitive and rapid detection, the principles of electrochemical detection, the nanomaterial enabled electrochemical detection. The reason why we'll talk about nanomaterial enabled is because the nanomaterial enabled are forming the generation Y kind of biosensors vis-a-vis the Gen X biosensors, which are simple nanobiosensors. The Gen Y biosensors, which are nanomaterial enabled, they offer a much higher sensitivity of response because of faster electron transfers. And since we are talking about electrochemical transduction, therefore we are talking about reactions which involve electron transfer. Therefore, these kind of biosensors can only be developed in cases in biological reactions which involve a redox reaction. Briefly gaining insights into a biosensor before we actually start talking about nanobiosensors and subsequently electrochemical biosensors. Biosensors will have a biological element in close communication with a transducer. The biological element is the one which is responsible for causing the biological reaction. That is why we differ a biosensor from a simple chemical sensor. A biosensor will evoke a biological reaction. These biological reactions can be evoked by certain biological recognition elements. These can be either enzymes or whole cells, tissues, antibodies, or aptamers. When we are looking at a highly specific response, we will be looking at antibodies and aptamers because antigen and antibody interactions are by far the most specific kind of interactions in which there is no crosstalk. Enzymes also offer specificity to the reactions because a certain enzyme will be active only for a certain substrate. Let's say if we want to detect glucose, so the compatible enzyme will be glucose oxidase. Or if we want to detect urea, the compatible enzyme will be urease enzyme. Whole cells sometimes have the ability to offer higher sensitivity of response because of their inherent capacity to produce enzymes. So rather than using a pure form of an enzyme, some enzymes are also expensive. So to give it a certain edge of affordability, we can also use whole cells vis-a-vis pure enzymes. We've also learned before this that the risks envisaged for say food safety could either be incidental or they could be intentional. Either incidental or intentional, both forms will eventually evoke a potential risk in food safety. The incidental risks classified mostly as microbiological risks which are present in food, and they can be imparted by the presence of Salmonella, E. coli, Listeria, Campylobacter, and Shigella. Other than this, the chemical risks which are mostly present or found in foods are classified as either contaminants. When we talk about contaminants, they can be chiefly ochratoxins or fumaricins or kanamycins, or they can be inorganic metal toxicity such as mercury and arsenic toxicity, which finds its way into humans through the consumption of contaminated fish. Antibiotic residues. Antibiotics find their way into human food by injecting animals by these antibiotics. One of the contact materials which is now on the list of WHO as a health concern is bisphenol A, which is present in the plastics. It is a hormone inhibitor. Another kind of contaminant which can be present, or a residue which can be present as a huge chemical risk in food, is the presence of pesticides. These pesticides could either be insecticides, herbicides, fungicides, or rodenticides. Fraudulent adulterants unfortunately have found their way into the food chain. The two most common fraudulent adulterants are the presence of urea and melamine in milk. Urea is used in milks which is synthetically produced. It is one of the ingredients for making synthetic milk and therefore acts as a biomarker for detection of adulterated or synthetic milk. Melamine, as we have also briefly learnt before, is a highly nitrogenous compound which imparts a false protein positive to milk. So if we put melamine in milk, it will give us a very high protein content. Therefore, urea and melamine are two fraudulent adulterant concerns in terms of food safety. Shown here are some birds which have consumed peanuts impregnated or laden with mycotoxins, and this is just to show that it's not just the humans which are affected by consumption of contaminated food, but it's the entire ecosystem which gets affected. Talking about food risks, food scientists the world over as well as food technologists are trying to develop techniques which are sensitive enough to detect these food contaminants and toxicants. The conventional food analytical techniques are time intensive, they suffer from cross interferences, and there is extensive sample pre-treatment required, which means that if we want to detect an antibiotic in a food, we will first need to extract that antibiotic by extensive treatment, and only then we will be able to detect that antibiotic. This has led to a new generation of sensing techniques which is called biosensors, the Gen X biosensors, and a step ahead where we use nanomaterial enabled biosensors which are called the Gen Y biosensors. In the Gen X biosensors, the advantages are that of a rapid response, the specificity of response, and there is no extensive sample pre-treatment required. As against a Gen X, which is a nanotechnology enabled biosensing device, which uses nanomaterials as one of the elements in the sensing element. The sensitivity of response in the Gen Y biosensors is far more than the Gen X biosensors, which means they can afford ultra-sensitive detection of analytes. Other than this, they are also rapid and they can be used for on-site detection. Transduction mechanisms are ranged into electrochemical, thermometric, piezoelectric, optical, and field effect transducers. Electrochemical biosensors form the realm or understanding of this module in particular, and these kind of transduction mechanisms are further divided into amperometric or imperimetric kind of biosensors. Thermometric transducers are those which detect a certain change in temperature of a reaction. Let's say we have a biological reaction going on in a vessel and it is accompanied by a change in temperature. If it is an exothermic reaction, there will be a heightened or there will be an increase in temperature. That kind of a transducer is called a thermometric transducer. So indirectly, by the degree of change in temperature, we will be able to detect the concentration of the analyte. Piezoelectric biosensors are physical biosensors. We have also seen in certain previous modules the way the cantilevers are developed. So let's say it's like a swimming pool board. We have a small board. We have an antibody, let's say here. So antibody sits on the edge of the board and its frequency of oscillation changes. But when an antibody comes and sits and makes an antigen-antibody complex, the frequency of oscillation will go down. So the rate at which that frequency of oscillation has fallen down will be indirectly telling us the concentration of the analyte. Optical biosensors are those in which there are lights, there is some sort of light response. That light response can be fluorescence, can be Raman scattering, can be plasmon resonance, or it can be simple changes in the spectra properties. Field effect transducers are those which aid in miniaturization of these devices. So in the lab, initially when we are developing bulk devices, we will be subsequently able to miniaturize those devices by the use of FETs. In this module, we will particularly focus on electrochemical biosensors. When we begin to understand about electrochemical sensing techniques, we will first understand what is the setup for this electrochemical response. So we will see that what is an essential component of electrochemical transduction is a 3-electrode cell. When we are doing experiments in a laboratory, we will have a small vessel in which there are 3 electrodes impinged. These 3 electrodes are the working electrode, the auxiliary electrode, and the reference electrode. Most commonly used reference electrodes are the silver-silver chloride electrodes. But the heart of an electrochemical biosensor or an electrochemical sensor is the working electrode. It is on the working electrode that all the sensing of the analyte is going to happen. When we talk about sensing of the analyte by the working electrode, these kind of sensing could be labeled as well as non-labeled. When we move a step ahead towards generation Y biosensors and we add nanomaterials in the working electrode, we are improving the response of working electrodes, which means, let's say, we have a flat carbon electrode which has not got any nanoparticles embedded in it. When an electrochemical reaction happens, there will be generation of current. Generation of current is essential because we're talking about redox reactions and hence electrochemical responses. We will also see the cyclic voltammogram in which there's a graph, there's an oxidation peak and there is a reduction peak. When we talk about nanomaterial enabled biosensors, we are putting certain nanomaterials and making them a part of the working electrode. By making them a part of the working electrode, we have enhanced the electrical conductivity of the working electrode. What does this mean? When we enhance the electrical conductivity and the redox reaction, biological reaction happens, the rate of electron transfer is much faster. If the rate of electron transfer is much faster, it only means that we are attaining a rapidity of response. If we are able to attain something in, let's say, 3 minutes, we will now be able to do it in a matter of a few seconds. Since we know that electrochemical biosensors are by far one of the most successful biosensors which are being used, let us understand now the concept behind the development of these electrochemical biosensors. Typically, an electrochemical cell is a 3-electrode cell. On the figure on the left, we will see 3 electrodes. In the middle, there is a working electrode. On the left of it is a reference electrode. And on the right of the working electrode is a counter electrode. And the figure on the right above shows the actual what we do in the laboratory. When we translate this 3-electrode cell onto a chip, it takes the form of what is shown in the figure on the right below. It has a 3-electrode system, which has in the center, the round is a working electrode, on top of it the counter electrode, and on the side of it is a reference electrode with their connections at the base. In the previous slide, we saw how a screen printed electrode is fabricated, and in this we see the steps for fabrication of a screen printed electrode. Slide 1 is at the base, which is usually a silica or a glass slide. Slide 2, wherein we have used a reference electrode. The most commonly used reference electrode is the silver-silver chloride electrode. Step 3, we etch in the working electrode. We will come to the importance of a working electrode a little later. Step 4, we embed a counter electrode just on the side of the working electrode, and then we screen print it, which becomes a screen printing electrode. The output signal in electrochemical transduction can be either electrical conductivity or resistance or impedance. When we have to analyze an analyte in a solution, what is shown in the picture above, we use the screen printed electrode, place a drop of the solution on the working electrode, And what we will see is a response called a cyclic voltammogram, which is depicted in the kind of graph which is shown in the image. The graph is a cyclic voltammogram on the x-axis of which is the potential and the y-axis shows current. As we scan the potential from the left to the right, there is one peak which appears, enhances. It is called the oxidation peak. And when we revert this cycle, there's a peak which comes below, which is called the reduction peak. So a typical cyclic voltammogram looks like this. It will have an oxidation peak as well as a reduction peak. The working electrode is the heart of a biosensor. It is on the working electrode that the biological reaction will happen and it will be detected. On the left, the image on the left shows a response of an electrochemical biosensor, and if you compare it with the figure on the right, we will see the appearance of distinctive peaks. This figure is just showing an ineffective biosensor working electrode on the left as against a more effective working electrode placed on the right. A more effective working electrode would typically mean a well-fabricated, electrically conducting working electrode. When we say a well-conducting working electrode, this is what we mean. What is shown here is a very distinct oxidation as well as a reduction peak. I would like to take your focus only on the CNT portion of the peak, which shows a very distinctive oxidation as well as a reduction peak in comparison to the graphs which are shown on the left. The reason why we have got such distinctive peaks is by the use of carbon nanotubes in the working electrode. Carbon nanotubes are known to have very good electrical conducting properties and therefore they enhance the response of a biosensor. When we say enhancing the response of a biosensor, what would it mean in food analytics? Shown in this graph are two images, two cyclic voltammograms. The one which is flatter is a flat carbon electrode which has been used, and one which with very distinctive peaks is a flat carbon electrode but with gold nanoparticles inside of it. Gold nanoparticles again are very good conductors of current, therefore they enhance the response. This means that at the same concentration of an analyte, a flat carbon electrode will not give us so much magnitude of current, whereas a carbon electrode which is impregnated with gold nanoparticles will give us very significant peaks. When we understand this at a different level, it only means that if we are trying to do ultra-sensitive detection of an analyte, and if we have used nanomaterials in it, highly conducting nanomaterials, we will be able to detect these analytes at ultra-sensitive concentrations, which is the need of the hour for food safety. Now that we have understood the use of highly electrically conducting working electrodes in electrochemical biosensors, here are certain more examples of more materials which are also electrically conducting. On the top left is a graphene sheet. When we use graphene sheets along with the working electrodes, it also enhances the response, the current responses manifolds. On the top right, we see carbon tubes or rods, carbon nanotubes or carbon nanorods. Such kind of arrangements also enhance the electrical response. And the figure shown below, it shows zinc oxide nanoparticles, but on the right, it also shows a nanocomposite, wherein is a combination of not only zinc nanoparticles, but on top of that are also placed zinc oxide nanorods, which will further enhance the response of a biosensor. So what did we learn till now? We learned that electrochemical transducers are applicable only if the reactions involve a redox species. A 3-electrode cell comprising a working electrode, a reference electrode, and an auxiliary electrode are essential for effecting an electrochemical response. The working electrode is the heart of the electrochemical biosensor, and nanoparticle embedded materials enhance the response of biosensors, which leads to ultra-sensitive detection of analytes. In subsequent slides, we will see certain specific examples of electrochemical nanobiosensors developed for food safety applications. Let us now understand a few specific examples of electrochemical nanobiosensors which have been developed for food safety. The first example which is being put forth to you is a sensitive electrochemical detection of a pathogen Salmonella with chitosan gold nanoparticles composite film. In this case, the working electrode is a chitosan with gold nanoparticles embedded in it. The advantage of using gold nanoparticles is because of their easy functionalization. In this example, what is shown here is a surface functionalization of gold nanoparticles with an amino group. When with the presence of these amino groups is the possibility of attaching an antibody to the gold on the surface of the gold nanoparticle. This antibody is the antibody against Salmonella. So when a Salmonella comes in contact with the antibody, an antigen-antibody complex is formed. This is followed by incubating the mixture with a secondary antibody which is functionalized with horseradish peroxidase. Now, horseradish peroxidase will oxidize hydrogen peroxide which is present in the system, which will lead to a redox reaction. It is important to note here that all electrochemical biosensing techniques will need redox active species as a part of their biological reactions. And if that is not so, then we cannot combine an electrochemical transducer with the biological system. Therefore, it is absolutely important to add an electroactive species. It is the presence of this electroactive species that will eventually lead to a response. What is shown here is a reduction current, only the reduction peaks. So with the increase in concentration of Salmonellas, there is increase in concentrations of HRP, and with increase in concentration of HRP, there is heightened reduction of hydrogen peroxide, which can be seen as reduction peak currents. This example shows the electrical percolation-based biosensor for real-time detection of Staphylococcus. Staphylococcus, which is yet another pathogen. Shown here on in the figure A is an electrically conducting film with black long fibers depicting electrical conductivity, in which there is passage of current between the electrodes. When there is excessive flow of electrons between the electrodes, the resistance is less, which is shown by the electrode. This electrically conducting film has got immobilized antibodies against Staphylococcus as sensing elements. So when this setup is incubated in the presence of Staphylococcus with the antigens or the enterotoxin B, which is a protein, it gets attached to the antibodies, makes an antigen-antibody complex, and reduces the flow of current, which means it enhances the resistance of the biosensor. This enhancement in resistance is in turn a measure of the concentration of Staphylococcus enterotoxin which is present. This example is of an electrochemical detection of Salmonella using gold nanoparticles. In this case, the gold nanoparticles are functionalized with the antibody, which forms the primary system, and these nanoparticles are magnetized. We can see the red horseshoe kind of magnet. So when this complex is brought in close contact with the magnet, they can be separated out. When this magnetic nanocomposite is taken out of the system and it is treated with a secondary antibody, this whole complex leads to enhancement in current merely because of the presence of gold nanoparticles. It is an indirect sensing mechanism wherein the mere presence of gold nanoparticles tells us indirectly about the complexation of the presence of Salmonella. The TEM image is shown. On the left, A is that of magnetic nanoparticles, and on the right, we can see few cells of Salmonella. Bisphenol A is one of the toxicants which has been identified by WHO as a health hazard. In this example, there has been the use of carbon nanotubes for ultrasensitive detection of bisphenol A. Bisphenol A is a serious concern for food contact materials in relation to baby foods. This is an example of a chronoamperometric urea detection in milk. In this case, the electroactive species is the material of the contact material, is the material of the working electrode, which is polypyrrole. Polypyrrole, in the presence of urea and urease, undergoes certain protonic changes, and these protonic changes lead to the electrochemical detection of the concentration of urea. Before we conclude our understanding of electrochemical biosensors, and though not in the direct realm of food safety sensors, I must reiterate here that the bioassay principle of glucometers, the commonly used devices, household devices for measuring blood glucose, is based on electrochemical sensing. The working electrode in that is a glucose oxidase, which is an oxidoreductase enzyme with FAD as the cofactor. It leads to the conversion of glucose into gluconolactone in the presence of an enzyme, and this enzyme undergoes a redox change, which is depicted electrochemically by the oxidation at the anode of hydrogen peroxide, leading to current generation. This current generation that indirectly gives us a concentration of glucose which is present in the system. Let us summarize this module. Initially, we learned in this module the very need for food safety biosensors. We spoke about food safety in terms of pathogen detection, in terms of chemical contaminants, and we classified these chemical contaminants into additives, contact materials, fraudulent chemicals, or certain metallic contaminants too, and the importance of analyzing these contaminants in food. The nanobiosensors, they offer sensing platforms that satisfy these needs. These nanobiosensors have an edge over conventional techniques, wherein the conventional techniques are not able to satisfy the responses in terms of ultrasensitive detection or in terms of rapidity. It is these nanobiosensors which will offer us to overcome the conventional techniques, the limitations posed by the conventional techniques. Electrochemical nanobiosensors are essentially those which involve redox reactions, which means there is an electron transfer happening in these reactions. Although we have used a biological system, it's a biosensor, that biological system evokes an electrochemical response, which is then detected by the electrochemical transducer. By adding nanomaterials into the working electrode, we are able to enhance the sensitivity of detection. We are also able to add the rapidity of detection because nanomaterials are highly conductive. We have seen examples of graphenes and nanorods and nanoparticles, which once embedded into the working electrode enhance the rate of electron transfer and hence increase or improve the response time manifolds.